Alright, let's talk about the two different types of specific immunity. In this video, we're going to talk about humoral immunity, and then our next one we'll go ahead and talk about cell-mediated immunity. Well, humoral immunity is part of our specific line of defense, and it's based on our B cells. We know that the B cells, they differentiate in the bone marrow, so this is where they mature in the bone. That's where they got their B cell name from. And they contain these antigen receptors that basically what they're looking for is their foreign counterpart. So if this is a B cell for a certain virus, it's going to be able to look at the antigen for that virus and be activated anytime that virus is in the system. If not, then it sort of just sits in stasis. The antigen is basically, and that's what this is kind of talking about, that there's one B cell for every antigen out there. So for virus 1, there's a B cell, and the antigen would be virus 1. For virus 2, there's a B cell, and it's a completely different one. And then for virus 3, there's another B cell. And we can go through this with bacteria and viruses and parasites, a whole bunch of other things. But anything that your body finds foreign and beats up once, it makes a B cell for it and a T cell for it. And so your body could have potentially millions of these different B cells, and they wait in your lymphatic system. They do mature in your bone cells, but they hang out in your lymphatic system, screening blood, looking for these foreign bodies, and when they find them, they become active. So let's kind of read and see what they do. So when one of them gets activated, what happens is a foreign molecule will have an antigen on it. The antigen is presented through this MHC2 complex to a T helper cell, which we'll talk about soon, which is going to release a bunch of chemicals that stimulate B cells to differentiate and to multiply. So let's say that this MHC2 complex, this white blood cell MHC2 complex, has activated a helper cell, and it's the one for that specific virus, virus number 1. It comes to the B cell for virus number 1. It starts to multiply. Then the B cell is going to become two different types of cells. It's going to become a plasma cell and a memory cell. But before it does that, it's got to kind of do this thing first. It's got to do this thing called clonal expansion. And clonal expansion is where the B cell starts to replicate, quite a bit of them. And some of those new cells, when it's cloned, will become plasma cells, and some will become memory cells, and some of those B cells will float around and go to other lymph nodes to activate them. The plasma cells are going to make antibodies that are specific for that item, and then when they find them, they're going to go ahead and attack them. So let's look at the picture. So here are some foreign bodies, and we go ahead and get them into the cell. We're going to go ahead and put those foreign bodies on the outside. That's going to be an activated MHC2 complex. And what that's going to do, it's going to have a B cell and a T cell. The T cell sort of activates this guy in this reaction. And what will happen, there's sort of a little step missing, that basically the B cell will start to clone itself. Some of those clones will become plasma cells, and some will become memory cells. The plasma cells will go ahead and start making antibodies. Remember we said that the most efficient ones will go ahead and make up to 1,000 in 60 seconds. And remember, these are just around to float around, find whatever those foreign molecules are, tag them, and then white blood cells or something else will come and kill them. The primary and secondary humoral response is going to be that we're going to have those IgMs, those immunoglobulins M's and IgG produced. And what they'll do is they'll sort of be in low sort of numbers. Typically you get sick and then you feel better. It's because you're making lots of memory cells and you're making a lot of antibodies, and they're sort of killing those items off. And the second response later will be if you see it again, you can kind of kill it off. So let's look at this process. So what's happening here is, so let's say this is day 0. That's when you're infected with whatever this is. And what happens is you'll see that you're getting sick, you're sort of feeling malaise, your body is starting to figure out what this is, and it's going to start to make IgM and IgG. And as we start to go through days, somewhere around here is where you're going to start to feel better. And your body is definitely going to be beating it up. And it's because these IgM and IgGs, right around here. What we're going to do is continue making these for a while until we know that we've completely killed all those items. And then somewhere around day 20 or so, we're going to completely end that process. So now let's say in the future, it could be day 2, it could be whenever, you have a second exposure. This could be 5 years later. What will happen is IgG and IgM will ramp up in high numbers and deal with it so quick that you won't feel any symptoms. If you do, it might be that malaise sort of feeling that you get when you feel run down, and that'll be it.